Once again, CBSE class 12 biology. In the previous class, we are discussing about the post-fertilization event. First part of that is the development of endosperm. Now the second part of that is the development of embryo. The process of development of embryo is called embryogenesis or embryogenesis. So today we are going to discuss about the embryogenesis. Now what happens when the endosperm is formed? And secondly, two nucleus, one is the male, one is the female, having n number of chromosomes. Male, female, having the n number of chromosomes fused together, combined together to form a diploid structure having two ends, and this we call zygote. And the process is called syngenemy. Syngenemy is the process by which the zygote is. Now what happens next? This zygote goes on in developing stage. But must remember, the development of embryo is started soon after the formation of endosperm, because endosperm is the nutritive part of the embryo. Embryo gets nutrition during the developmental age through endosperm only. So after the development of the embryo and development of endosperm, this development starts. This is the zygote. Zygote further divided into two parts. Mitotically divided to form two parts, and this is called proembryo. Proembryo. Now two parts are there. One is the spun and one is the spun. This is the microphylar part. This is the terminal part. What happens in this part? We are studying it separately. Anyhow, the process going on simultaneously. This is the microphylar part. This is the chelaisal part or the terminal part. What happened here? This first cell enlarges and we call it hostorium. This is hostorium. Hostorium further develops number of cells here, that is 6 to 8 cell structure. This is 6 to 8 cell structure developed by the hostorium. And what is the role of this structure, 8 cell structure? It is actually filamentous. Okay, it is filamentous structure. Filamentous structure, and we call it suspensor. Suspensor. The last cell of this suspensor is called hypophysis. Hypophysis. Now, what is the role of suspensor? Suspensor is actually the part which is inserted in the endosperm. It pushes the embryo in endosperm. Okay, it pushes the embryo in endosperm. Why pushes the embryo in endosperm? Because endosperm is the source of food for the development of the embryo. So this is 6 to 8 cell structure. 6 to 8 cell structure. And what is important here? It is filamentous, suspensor, and is suspended inside the endosperm because it is the source of nutrition. Or in other words, we are able to say that the embryo gets nutrients from endosperm through suspensor only. Now what happens with this cell? This cell present in the chelaisal end or terminal end. This end is called the terminal end. Okay, what happens with this cell? The cell which is present in the terminal end. This cell is called embryonal initial cell. Okay, what is this cell is called? This cell is called embryonal initial cell. Initial cell, it means it is going to form. It is going to form the initial embryonic cell. What happened with this? This embryonical initial cell, this embryonical initial cell which is present near the hypophysis. What is the location of this? This is the terminal end. This develops to form hostorium, suspensor, and the hypophysis. At the end of the hypophysis, I'm going to write the name embryonal initial cell. Okay. Now, what happens with this embryonal initial cell? This embryonal initial cell enlarges and goes in the division state. What is the division state? It divides mitotically to form 2 cells, again to form 4 cells, then to form 8 cells. It means it reaches to the 8-cell structure. 8-cell structure. So out of this 8 cells, 4 cells are present in the lower tier and 4 cells are present in the upper tier. All the cells which are present in, this is embryonical cell divided into 8 cell stage. This is the place that is hypophysis, where it is added to the substance. Hypophysis. Okay, what happens with this 8 cells? At this stage, it is divided into 2 parts. One is called the lower tier, 4 cells present in the lower tier, and 4 cells is present in the upper tier. Okay, this lower tier of 4 cells, and these are the upper tier. Again, it also has 4 cells. In future, this lower tier cell is developed to form hypophysis. What is the role of this? It goes to form hypocotyle, and hypocotyle further develop into radicals. Radicals means it is going to develop the root. What is the meaning of the radical? It is going further for the development of the root. This is the role of lower tier. What is the role of upper tier? Upper tier also has 4 cells and it is going to develop in future plumule and cotyledon. Cotyledon. It may be 1 or 2. Okay, it is going to develop the cotyledon. It may be 1 or it may be 2. If it is 1, it is called monocotyledon, and if it is 2, then it is called the dicotyledon. Up to this, the development of embryo is common both in monocotyledon and in dicotyledon. Okay, so what happens? Look at here. First, the zygote is formed. The process is called syngamy, and whole process is called embryogenesis or embryogenesis. The development of embryo, first the microphylar end is going to divide. Microphylar end divides further to form suspension, haustoria, and hypophysis. Hypophysis is the terminal end where the chalizial cell is there, and the cell of the terminal end is called proembryonal initial cell. Okay. At this time, the zygote is called proembryo. Okay. What happened in the proembryo? The terminal end, the cell present in the terminal end, further divided mitotically from 1 cell to 2 cells, then 2 cells to 4 cells, and 4 cells to 8 cells. 8 cells, the upper tier and lower tier. Upper tier contains the 4 cells and lower tier also contains the 4 cells. The upper tier is responsible for the development of hypocotyle. Hypocotyle further develops to form radical, then the parts of the root cap, etc., etc. And what is the role of the upper tier? Upper tier also has the 4 cells. Upper tier further divides to form plumule, and plumule further develops the plant or the whole shoot system with the formation of one or two cotyledons. Up to this, the development of embryo is same in monocotyledon as well as in the dicotyledon. Now let's move. Okay, let's write here. Up to this, the development of embryo is common or same. In monocot and dicot. Here, up to here, the process is same. And this process is called, up to here, the 8-cell structure is formed. 8 cell of the suspensor as well as the 8 cell of the terminal end or the embryonial initial cell. What we have to write, I'm going to rub this. Okay, now what is going on up to here? Must remember, up to here, the process is very much common for monocot as well as the dicot. So what happened up to here? First, the process of development of embryo is called embryogenesis. Embryo develops from micropylar. Okay, micropylar, it develops, start development is the micropylar end, and this development is started soon after. Development is started soon after the formation of endosperm. Then what happens? The micropylar end divides to form suspensor, that is 6 to 8 cell filament is formed, which pushes the embryo in endosperm, because endosperm is only source of nutrients for the development of embryo. At the same time, the terminal end also divided. Terminal end developed into embryonial initial cell, divide mitotically to form 8 cells. And you know that 8 cells is further divided into lower tier and upper tier. You must write here. The lower tier is responsible for formation of hypocotyl and the radical root cap part there. And the upper tier is responsible for formation of the plumule and 1 or 2 cotyledon. Up to this, the development of embryo is common for both monocot as well as the dicotyledon. This is the development of dicot embryo. Okay, now this is the development of dicot embryo. Okay, I am going to repeat the process same. What happens? Zygote is there. This is zygote. Zygote developed to form pro-embryo. This is zygote. Zygote developed to form pro-embryo. Pro-embryo then divide hausturium, suspensor, okay. Here the 8-cell structure. And this time it is called globular embryo. Globular embryo. I'm not going to label it again because you know very well whatever the parts are. This is called hostorium. This is called hostorium. This is suspensor. This is hypocotyl, and this is the group of two tiers, that is the lower tier and the upper tier, which is responsible for the formation of radicle and the plumule. Okay. Now what happened next? The development of this part, this whole together is called the globular embryo. The development in this upper tier and lower tier goes on continuously. In several planes, they develop continuously, and because of that development, their structure changes. Okay, this structure gets changes and it is somehow heart shaped in structure. Number of cells increases now because it divides continuously in same plane. The division is mitotic division. Number of cells increases and the shape becomes heart. So it is called the heart-shaped embryo. What is this? This is the heart-shaped embryo. This is heart-shaped embryo. Okay, up to here, the development is common, both in monocot and dicot. But from here, the development is little bit changed. This part is called the globular embryo. This is zygote. Then zygote gets converted into proembryo. Proembryo gets converted into globular embryo, and then heart-shaped embryo. Okay. At this time, the division is going on continuously for the formation of the cotyledon. Now, what happened next? This is the part which is suspended inside the endosperm because the development process is going on. It is not stopped still yet. So what happens? This part divides continuously. First, it forms the heart structure, but afterwards, it forms the complete structure, somehow the shape of horseshoe. It divided continuously to form somehow the shape of horseshoe. This is the two cotyledons. Okay, these are the two cotyledons. Two cotyledons are formed here. Okay, this is the two cotyledons. This structure is formed. This is the terminal end. Again, this is the microfilar end. As same, you know that. What is this? The development is going on here. And at this time, this is the terminal end. This is the terminal end. This is cotyledon, two cotyledons. Two cotyledons, because this is a development of dicot plant. This is the responsible for the development of radicle. This is responsible for the development of plumule. Okay, this is actually the part where the plumule is formed. Development of plumule. This change is going on in dicotyledon plants, not in the monocotyledon. It is specified for the dicotyledon plant, and it is near about horseshoe in shape. Horseshoe in shape. Okay, this is the complete development of dicotyledon embryo. So what happened in the dicotyledon embryo? The upper tier and lower tier divide in several planes again and again and form the heart-shaped embryo. Okay, a heart-shaped structure that is called heart-shaped embryo. Then it divides again and again to form one horseshoe-shaped structure in which the plumule is present, the radicle is clearly visible, and two cotyledon is also clearly visible. Okay, hypocotyle is there, hypocotyle enlarges. This hypocotyle is there, this part is the hypocotyle, this enlarges to form the horseshoe shape. And this is the complete development of dicot embryo. So this is the development of dicot embryo. What happened here? The process, complete process is zygote, forms proembryo, proembryo develops into global or globular embryo. Up to this, the process is same in monocot and dicot. So we have to take the separate process that is going on here. Embryonal axis is developed. Embryonal axis is developed. Okay, this heart-shaped embryo, the two end is there. One is this, and the second one is this area. This is called hypocotyle. This one is called the epicotyle. Hypocotyle develops to form, from the lower tier, to form radicals, root cap. Okay, what is the role of epicotyle? Epicotyle develops to form plumule. Okay, it develops to form plumule. This is called the mature embryo. It's horseshoe in shape, and this is called mature. So this is the embryonic development in dicotyledon, the structure of a developed embryo. This is the typical dicot embryo. Typical dicot embryo. As we see that, this all are called the embryonal axis. This is epicotyle. This is hypocotyle. Hypocotyle is the lower end, which is responsible for the formation of radicals, root and the root cap, and the epicotyle is the upper end. It is responsible for formation of the two cotyledons, which is clearly visible here, and this is the plumule, the further develop the shoot. So this is a typical dicot embryo. Now what happens with the monocot embryo? Monocot embryo, in your textbook, the embryo that is given is the TS of the grass embryo. This is the TS of transfer section of grass embryo. Grass embryo. Grass is monocotyledon, so it is a type of the monocot embryo. What happened in the monocot? Only one cotyledon is there. This is called the scutellum. It is present in this end. This is a terminal position. Single cotyledon is there. Okay, the scutellum at terminal end. At terminal end. What happened at the terminal end? At the terminal end or terminal position, the single cotyledon is present, and that single cotyledon is called scutellum. At terminal end, single cotyledon is present. Cotyledon, this is a single cotyledon, is present, called scutellum. Okay, it is a scutellum. At that end, that the lateral position, the plumule is present there. Okay, this is somehow the shield in shape. The shape of scutellum is shield shaped scutellum. Okay, this is the part which further develops into the Plumule. Plumule is developed from the lateral position. Plumule developed at lateral position. Lateral position is clearly visible there as a shoot effect. Plumule protected by a cover. The protecting cover which is present over the plumule is called colectile. A protecting cover is present on plumule called colectile. Okay. What is the next? Radicle is also protected by some layer. The radicle is protected by this layer, and this is called chorioryza. This layer is called chorioryza. A protecting cover also present on radicle called chorioryza. Okay, this is the typical monocot embryo of the grass. This is the embryo of the grass family. And ultimately, what happens? Ultimately, the two things is going on. Ultimately, as the result of both monocot and dicot. This is in both monocot and dicot. Ultimately, what happens? Seed is formed, ovule get converted into seed, and ovary get converted into. Okay, our next topic is the formation of seed and the